Kusasampula and welcome to our DNA module prepared by Master in Education Biology second year. These are the team members. Deoxyribonucleic acid, popularly called DNA, is the genetic material which is present in all the organisms, and then it carries the genetic information from parents to offsprings to next generation. Any developmental activities that's taking place in our body, growth, reproduction, susceptibility to disease, these are all being taken care by the genetic material which is present in the DNA. We have prepared a model of the DNA. So this is how a normal DNA would look like. They are composed basically of three main parts. The white part which we try to present here is the skeleton, the backbone of the DNA, which is made up of phosphate and sugar. So at every sugar ends, then they are being connected by nucleotides of which are made up of 4 bases. So the 4 bases are namely adenine, thymine, cytosine and guanine, which are represented by the capital letters A, C, G and T. Now if you look here, we have tried to bond these bases together. So a pair of bases are being attached here at the sugar skeleton. So for every thymine, it attached with another base, that is adenine, and then for every guanine, it attached with the cytosine, and then at a point where they attach, they are being bonded by hydrogen bonds. So this is how the DNA. Now in human organisms especially, we have 23 pairs of chromosomes, and so we are also supposed to have 23 pairs of DNA, each chromosome in each DNA. Now with the advancement in technology, scientists are able to make complete modification to DNA of an organisms. This they do by different tools and techniques like recombinant DNA, PCR, and then gene editing tools like ZFN, TALENs, and then CRISPR-Cas9. What they do here is we have tried to present how gene editing is done in the DNA by this another still model. So if you look very closely, it is the same DNA, but then we have tried to modify, make changes to the sequence of the DNA here. So this sequence of the DNA in a DNA strand is called as genes. So according to what the scientists want to do changes to the gene, then they can modify them accordingly. For example, if the scientists want to clone part of this gene, so what they will do is they will take out this gene and then make modification accordingly. Either they want to multiply this clone and then make multiple copies of this gene which have a desired characteristics to be used for different purposes. Or let's say in the genetic makeup of an organism has a disease. So then by analyzing, studying the genetic makeup of the DNA, scientists would be able to find out which part of the DNA needs to be replaced. So accordingly, they can make changes here. Take an example in sickle cell anemia, one of the most common blood disorder in the US. What happens is, as in the 6th codon, so when I talk about codon, it is about the arrangement of these bases. So a group of 3 bases forms one codon. So what happens? First codon, second codon, third codon. In the 6th codon, the arrangement is supposed to be GAG, guanine, adenine, and again guanine, and then it forms the glutamic acid. But due to some abnormalities in the sequencing of the genes, in the place of adenine is being replaced by thymine. So instead of glutamic acid, what happens is valine is formed there. So because of this, the change in the structure of the blood vessel, our blood RBCs, the hemoglobin, it's supposed to be biconcave, circular, and then they're very smooth. But what happens because of this change from this base from A to T, then it leads to change in the shape of the blood vessels. Then they become crooked, and then in a form of a sickle shape. It becomes hard, sticky, and then it clogs there in the blood vessels, and this prevents the blood reaching to different parts of the different parts of the cells carrying oxygen. So these are some examples. Now, having known that which part of the genes there are deforms where they need to make changes, scientists can then work accordingly. They can remove that thymine, and then in place of thymine they can replace adenine again. But now what happens, this gene editing raises a lot of ethical issues. So far, no countries in the world allows this gene modification to human beings due to ethical issues. This is because in gene editing there is a high degree of precision, of accuracy in DNA sequence, and then any deformities, any abnormalities can lead to the mutation. So that's why it is not allowed in human, but then this gene editing has been done in producing genetically modified organisms like some fruits, vegetables, and then some other organisms like chicken and fish. So we have some examples of GMOs here. These GMOs are being used and consumed in some of the highly populated advanced countries, but not in our country. By law, it is not allowed here. Now behind me there are 3 chart papers. So here we have the DNA fingerprinting. This is one application of gene editing. Because of the improvement in science and technology, this gene editing, this DNA fingerprinting uses the concept of this gene editing in forensic lab. And then we also have this CRISPR-Cas9, an example of gene editing tool where scientists use it in the field to modify the genes about how the unwanted genes are being cut from the strand, and then they are replaced with new healthy genes here. Right in the middle, there is also another chart. So this chart, we are trying to represent the characteristics which are being carried down from generation to generation by this DNA. For example, if the father is, the parental genotypes here, if the father is tall, and then mother is tall, so how would their offsprings be like? In case if both the father and the mother are dwarf, how would their offsprings look like? So this is the Punnett square, and then accordingly we can calculate how the genetic characteristics in the parents can be transferred to the offsprings, and then so on. So this is all with regard to what we are trying to explain through this DNA and then gene editing. Thank you.